Hello students, a few of you had asked for a video about the electron transport chain, as it's possibly the trickiest bit of respiration, and it's good to be able to go over it again and again. So what I'm going to do is I'm going to use the slides that are on Godalming Online that we used in the lesson, but I'm going to talk through it in what I hope is as clear a way as possible, so that you can review and watch a few times until you get it straight in your head. Now, we're talking about oxidative phosphorylation here. We're also talking about the electron transport chain. So the phosphorylation bit, the oxidative phosphorylation, that's the actual making of ATP. And it's called oxidative because it's driven by a series of redox reactions in the electron transport chain. Okay, so what is the electron transport chain? It's a series of proteins embedded in the inner mitochondrial membrane. Now, all through the other parts of respiration, glycolysis, link, the Krebs cycle, we've been producing molecules like reduced NAD or reduced FAD, or NADH or FADH2 if you want to call them that. And we've just been making these and then saying they'll play their part later on. Well, this is the bit where they all come into their own. So some of these will have been produced in the cytoplasm during glycolysis, but they of course can be moved into the mitochondria. And a lot of these will have been produced inside the matrix of the mitochondria during the link reaction and the Krebs cycle. So there's lots of these in the right place already. And what they're going to do is they're going to deliver their high energy electrons to these proteins here in the electron transfer system. In the process, they are of course going to become NAD and FAD, so they won't be reduced anymore. This yellow arrow is representing what we'd call an oxidation reaction, because oxidation is loss. They've lost those high energy electrons. That means that the energy is being transferred to this system of proteins here, where it can be passed from one molecule to the next. The ultimate aim, of course, is that we're going to get some ATP out of this, but in order for that to happen, electrons have to flow through the electron transfer system until they arrive on the final electron acceptor. That's oxygen up here. Electrons are going to end up on that oxygen molecule, going to ionize it, so it gets a negative charge, and then of course it's going to want to grab some hydrogen ions, and that's where we form water, the waste product. ATP is produced in this process, and it's reckoned that you get about 3 ATP molecules for each NAD that drops off its electrons. And FAD, well, the electrons it's dropping off are not quite as high energy, so you only get 2 ATP molecules there. These guys, once they're reformed, NAD and FAD, they go back into the matrix, maybe back into the cytoplasm, but they're going to rejoin all of those other stages of respiration where they are required, like glycolysis, link, and Krebs. Okay, the idea is that NAD, or FAD in fact, they act as carriers. They can take on board high energy electrons, loading themselves up with energy effectively, which they can then deliver to the electron transport chain. Right, this is where it's happening. So this is our mitochondrion. Let's get a slightly clearer version. What we're really focused on is this membrane here, the inner mitochondrial membrane. Okay, so it's a phospholipid bilayer, just like all the other membranes we've been talking about, but it's highly folded to give it a large surface area, to give these infoldings here. And all the proteins that we look at are going to be on this membrane. When we talk about pumping hydrogen ions, we're pumping them from the matrix, which is blue on this diagram, into the intermembrane space. That's this gap here between the inner and outer membrane. Okay, it's an enclosed space, and we're going to be pumping some hydrogen ions in there. Right, this link is well worth watching. It's a video that goes through this in a bit more detail than I'm going to discuss. So after we've finished here, load this up on Godalming Online and follow that link. But I'm going to discuss it in the simplest possible terms. So this is the electron transport chain. So what we're looking at here is just a little bit of this membrane, zooming in. Okay, and this is the matrix down here, and then this is the intermembrane space. So here is our NADH, our reduced NAD, with those high energy electrons. It's arrived at the very first molecule, protein molecule, in the electron transport chain. Okay, it's a big protein complex that spans the membrane, and NADH is going to be able to give up its electrons. Okay. Oxidation is lost, so we would say that our NADH is being oxidized. There we go, it's gone back to just NAD, and the electron is now on this protein molecule. The electron is part of this protein molecule here. And of course it's a high energy electron. So when this protein molecule hands the electron onto the next acceptor, some of that energy is transferred to it. Enough energy is transferred to pump a hydrogen ion into the intermembrane space. This little molecule here, you don't really need to worry about it too much, it's just a carrier. It's going to hand that electron onto the second proton pump, and the same thing's going to happen. It's going to use that electron's energy to pump a hydrogen ion into the intermembrane space. Another carrier molecule comes along, it's handed onto a third proton pump, and you pump a third hydrogen ion in. So this is the same electron that came from this NADH molecule at the very beginning, but now it's gone through 3 proton pumps, it's got to the end, and it's transferred all of its energy. It's now exhausted in a way, and it needs to leave this molecule so that the electrons behind it can continue the flow, and it can only do that if there's a final electron acceptor to take it away, and that's this, oxygen. This will have come from the surroundings, so if you're a single-celled organism, it would have just diffused in. If you're a multi-celled organism like you and me, then of course it's been delivered by hemoglobin, and then it's diffused into the cell and into the mitochondria. But once this oxygen is here, the electron can move from this final proton pump onto the oxygen, forming water. And of course it picks up a couple of hydrogen ions to do that. The whole point of this is we've pumped lots of hydrogens into this intermembrane space. They're trapped in there. They cannot diffuse back through this hydrophobic bilayer because they're highly charged. So we're just steadily stacking loads and loads of hydrogens into this space. Now I know that these have got confusing Roman numerals on them. Do not worry about that. All you need to know is that there are 3 proton pumps. These guys are just carriers that help the electrons get between them. Okay, so don't worry about the Roman numerals on that at all. Right, let's have a talk about this one, FAD, reduced FAD here. You'll notice that reduced FAD also plays its role, but it doesn't do any interaction with the first proton pump. In fact, its electrons join partway through the electron transport chain. This is because it has only got electrons with kind of a medium to low level of energy. It doesn't have the super high energy electrons that NADH does. So this is only able to drive the second and third proton pump, and as a result, one molecule of FADH is only going to pump in 2 hydrogen ions. But that's still worth having. Let's skip ahead a little bit and think about what happens next. All of those hydrogen ions are collected in the intermembrane space. There's absolutely tons of them in there, and that's what we call an electrochemical gradient. They are going to flow down that gradient if you give them a way out. And that's what this molecule here looks a bit like a keyhole upside down. This is ATP synthase. This is a channel that allows hydrogen ions to flow through, and in doing so, that flow of hydrogen ions, just like water turning a water wheel, is going to drive this enzyme. So the hydrogen ions are not directly getting involved in this reaction down here. They're simply driving and providing the energy. But this enzyme is now going to be able to take ADP and a phosphate and bolt them together. So the hydrogens are not taking part in that reaction per se, they're just providing the energy. Now this is called chemiosmosis. The idea being that as long as you can maintain that nice gradient of hydrogen ions, you will be able to produce ATP in these stored particles, these enzymes, ATP synthases, on the inner mitochondrial membrane. Okay, that's what it actually looks like. There's a rotating bit in there. Okay, so how do you learn this? Well, you take a diagram like this. It's initially a bit shocking and a bit detailed, but actually once you study it, you realize this is not too bad. This is the matrix down here. This is the intermembrane space up here. And there are your 1, 2, 3 proton pumps. This is a reduced NAD going all the way through, powering all 3 pumps, and then going to oxygen. Reduced FAD, of course, is not on here, but it would join at this stage. So what you need to do in order to get really comfortable with this complicated process is get yourself so that you can draw this from memory. Okay? Go back, rewatch the video, and then have a go at doing that.